Welcome to S Chemistry. I'm your instructor Dr. Suhas Gauri and I will be teaching BSc 5th semester Physical Chemistry course on nanomaterials. Here is the outline. In this class, I am going to cover first synthesis of nanomaterial using microorganism, followed by synthesis of nanomaterial using fungi, followed by synthesis of nanomaterial using plant extract. So let us start with the synthesis of nanomaterial using microorganism. So how to synthesize using microorganism? So microorganism, as we all know, are the organism that can be observed under a microscope. Micro means the size of the organism is in micro scale, that is 10 to the power minus 6 meter. Uh, which are the micro which are the microorganism? So these three are the microorganism: bacteria, fungi, or yeast. So these three types of microorganism we are going to see now. So microorganisms are capable of interacting with metals, combining coming in contact with them through their cells and forms nanoparticles. So when this microorganisms comes with contact with any metal ions or metals, then they start absorbing that metal and using them. Um, they take the metal ions inside the cells, and that's how they form the nanoparticles. In some microorganism, metals ion enter into the cell, into the well body, and bind with certain proteins. Some microorganisms secrete a polymeric material such as polysaccharides which binds metal ion. So the use of various microorganisms can be given as. So some microorganisms produce hydrogen sulfide, that is H2S, and it can oxidize organic matter forming sulfate. So which is turn turns act like an electron acceptor for metabolism. So these three types of possibilities are there. That's how the microorganism synthesize the metal nanoparticle. So depending on the which microorganism is there, so these three types of mechanism is possible. So either your metal ion get enter into through cell wall and bind to the certain protein, and that's how it get interacted inside the cell. Or other is some microorganisms secrete the polymeric material like polysaccharide, and that these act as a surfactant, and that binds to the metal ions, and that's how it synthesizes the nanomaterials. Or some microorganism produce the hydrogen sulfide gas. So this is the oxidizing agent, where this sulfate get oxidized and form some this sulfate binds to the metal ion, and that's how it form the nanoparticles. So which it act like an electron acceptor for metabolism. This H2S can be presence of metal salt convert metal ion into metal sulfide, which deposits extracellularly. So now, using this table, we are going to understand what are the types or what are the species related to the microorganism and which nanomaterial they synthesized. So first we will think about the microorganism, that is the bacteria. So Pseudomonas statigeria AG25 lactobacillus. So this is the name of species of the bacteria, and that synthesize the silver synthesis of silver nanoparticle, that synthesize from gold or silver and alloy particles. So these are the lactobacillus and pseudomonas These two species are there in bacteria that is used to synthesize the silver nanoparticle or gold nanoparticle. Another species is Desaltobacteria says. So this bacteria species name is given here, and that is used to synthesize zinc sulfide nanoparticles. Another name is Klebsiella pneumonia. So this species is generally used to synthesize cadmium sulfate nanoparticles. So now we are going to see the second microorganism, that is fungi. So in fungi, this Fusarium oxysporum, Thermomonospora sp verticulum sp. So this species is used to synthesize a gold and silver nanoparticle or gold nano silver nanoparticle. So why is given? So generally the microorganism are living organism, and whatever the essential metal generally use this microorganism. So using that, only those metals are well explored by this methodology. So that's why the gold, zinc, cadmium, so these are the friendly metal to the microorganism. So only those materials are generally involved in the synthesis part. So another microorganism is the yeast. So Candida glabrata MKY3. So this species is used to synthesize cadmium sulfate nanoparticle and silver nanoparticles. So here are the few species and few nanomaterial synthesis are given here.
Moving to the next topic, that is synthesis of nanomaterials using fungi. So now we are going to see the synthesis of nanomaterial using fungi.
Now we will think of the particular example, that is the fungus of verticulum sp. So this using this fungus, how to synthesize the nanoparticle. So this should be grown in Elmer flask containing MgYp, maintained at 25 degrees Celsius. So Elmer flask is conical shape, uh, that if you store your fungus inside this flask, and you have to maintain the 25 degrees Celsius. The second procedural part is the flask is shaked for 4 days, and it is centrifuged at 10 degrees Celsius for 20 minutes. So this procedure requires 4 days, and 20 first you have to it is centrifuged at 10 degrees Celsius for 20 minutes after keeping for 4 days. So harvested myelilla mass exposed to gold nitride at 5 to 5.5 to 6 pH for 3 days. If you expose this is silver nitrate at this pH for 3 days, then you can harvest this mass whatever they have synthesized. So an Elmer flask at 20 degrees Celsius, verticulum biomass be fixed in glutaraldehyde in distilled water by maintaining it at a room temperature for 2 hours. So you have to add this glutaraldehyde into the distilled water, and you have to keep it at room temperature for 2 hours. After filtration, the centrifugation leads to all sedimentation rich in silver nanoparticles. So if you filter out this solution, and if you centrifuge, that will leads to the sedimentation, and that sedimentation rich in silver nanoparticles. So you can see the centrifugation instrument. So here you have to put the sample inside the centrifuge, and this two opposite two poles has to be weight balanced after putting this lid down, and if you close this lid, you have to rotate at some rpm, fixed rpm. So if you rotate at fixed rpm, due to centrifugal force, your sedimentation get at the bottom. So that's how we separate out the silver nanoparticles from the given biomass, verticulum biomass.
Now moving to the next topic, that is synthesis of nanomaterials using plant extract. In this topic we are going to see how to synthesize nanoparticle using plant extract.
Now we will see which plant, the plant extract, how to synthesize nanoparticle. So some plant extract can be used for synthesis of nanoparticles, leaves of geranium plant. So here we are going to see the plant species that we call it the geranium plant. So using this geranium plant, how to synthesize the nanoparticle. So first you have to take this plant leaves and we have to crush it well. The finely crushed leaves are put in aluminum flask and boiled in water for just a minute. You have to put these crushed leaves in aluminum flask and then you have to heat it in water for a minute. So this is the figure of aluminum flask. That's how it look like. So this is 250 ml aluminum flask. So leaves are ruptured and cell release intracellular material. If you crush the leaves, that will release the cell material, that is intracellular material, that we are going to use for the synthesis of nanomaterials. So solution is cooled and decanted. The solution, this solution is added to the H gold chloride, hydrogen gold chloride in aqueous solution. So you have to put this gold salt to the this aqueous solution. Then you will, when nanoparticles of the gold start forming within a minute. After addition of this salt, your nanoparticle will start immediately within a minute. So that's how we synthesize the gold nanoparticle using this plant extract.
So here we came to end of this chapter. So these are the references for the images where I referred, and these are the right hand side, these are the references for the book for this content. So finally I thank you all of you for kind listening. See you next lecture. Till then, happy learning.